here.
Okay, well, we can just get going because we have an hour kind of scheduled for this, and thanks for agreeing to do it, Mark. So I'll give a briefest of introductions. We all know Mark Churchland and his work, his pioneer in the domain of motor cortical physiology, and specifically working in monkeys on reaching and grasping, and I'm trying to understand the underlying neural dynamics that give rise to these kinds of actions, and has pushed our field forward in various ways. Today, he's going to tell us about kind of different way of thinking than the previous, I mean, related, but a different way of seeing a similar issue as before with respect to the flexible control of motor units. So just so everyone knows the idea, Mark is going to more or less give a 40-minute presentation, 45-minute presentation, and then we'll ask questions. But I assume you're okay if there's something pressing, we can hop in and ask you.
Yeah, absolutely. You know, and I greatly enjoyed the opportunity to be introduced by you and give a really good talk a couple of weeks ago. This will not be a really good talk, and there's some risk that it'll run over, but I can always we can always skip the end. But yeah, because there is a risk that it may run over, let's not have extended discussion during the talk. But absolutely, if you have a technical question or a question about a controller, whether something was done the right way or an important interpretation question, yeah, definitely press me on that, and I'll be happy to answer as I'll pretend to be Nausea and do my best.
Okay, awesome. Well, take it away at your leisure.
Okay, great. So let me share the screen.
Share. He checked. He should be able. Yeah.
We're working. It's working, right?
Yeah.
Okay, and you can see my
Yeah.
cursor as well. Okay, yeah, that'll be useful. Okay, so the work I'm gonna talk about today is the work of sorry, give me one sec here. There is some issue with it not wanting to. Well, that's okay. Apparently I just have to click. It won't work the normal way. It's just some weird Zoom / keynote interaction. Okay, work of quite a few different people and also spanning multiple labs and multiple advisors. But you know, first and foremost, this really was Nausea Marshall's project. He's on the left there. But also with some important heavy lifting from Josh Glazer, Eric Troutman, and Elam Amamastro. But really, what I'm about to describe to you is Nausea's vision in a lot of really sort of key and critical ways. So we're going to start with the observation, kind of obviously, that we produce a great variety of different movements in our daily lives. But of course, all of those are driven by muscles. And I'm going to give a quick refresher that probably nobody in your room needs, but it's in the talk, so we'll go through it anyway. Right, you've got motor neurons in your spinal cord. A given motor neuron uniquely innervates a subset of fibers within a given muscle. When it spikes, the neuromuscular junction is essentially perfect. So one spike in one motor neuron leads to one and exactly one spike in all of the attendant muscle fibers. And so because of that sort of perfect relationship, we refer to this as a motor unit. It means you can actually effectively be recording the spikes of motor neurons from the muscles, which is very convenient. A group of such neurons is called a motor neuron pool. That's all the neurons that go to a given muscle. And there's not a lot of them. They're sort of on the order of a few hundred for a largish muscle and only a few dozen for a small one. And they're controlled from both subcortical, principally brainstem, and cortical structures, with cortex making the largest contribution, especially in primate. So motor cortex itself is about 1 billion neurons, and the total cortical descending tract in humans is a little over 1 million axons. And they're controlling not a lot of motor units. There's only about 50,000 motor units that those million descending connections are controlling. So the central questions of this talk is really what's the nature of this control? How does neural control of muscles work? What can it do? What can't it do? What's possible? What's not possible? So to get into that, we're going to have to quickly go through some sort of present dogma. So with regard to cortex, or really the brain in general, the idea is that it exerts some level of control that's sort of at or probably above the level of the muscles. So you might have, you do have many hundreds of muscles, but the idea is many of them do similar jobs. And so a relatively modest number of synergies might be all you need to control the entire body. And this idea has been expressed in a variety of different ways, some more fanciful than others, but all of them have at their heart this important idea that I want you to keep in mind. By the way, is my audio good? I can't really see thumbs, so you have to say something.
We are muted.
Yeah, maybe
Are we
he is.
muted?
No, we shouldn't be.
Now you're not. So is the audio good, or are there any
Yes, it
adjustments?
is very good.
Okay, great. So yeah, sort of sear in your brains this idea that what's important about the synergies is that they are said to simplify the control problem from the standpoint of cortex, right? They relieve cortex of the burden of controlling each and every individual muscle. There's a similar idea at the level of the motor units, but we're going to get to it in a bit more of a roundabout way. So motor units are diverse in a variety of ways. For now, I'm just going to describe one way that's probably familiar to most of you, which is that there are large motor units. They're large because they innervate many fibers, so they produce big impulse responses for every spike. Typically, they tend, although they're not always, but they tend to also be innervate fast twitch fibers. So they get a big and relatively short impulse response. And in contrast, a small motor unit, one motor neuron and the fibers small number of fibers that it activates will, of course, get a smaller peak force, but then usually those tend to be slow twitch fibers. And so it's also a broader, slower response as well. And generally speaking in life, if you're controlling a diversity of different things, there's different trade-offs, and optimality requires respecting what those trade-offs are. So what do I mean by optimality? I mean, you have some target force you want to create, where you are the brain, and then there's going to be some force on the right that we're going to hope actually matches the force on the left. And what have you got to work with? Well, you've got these twitch responses, some big on the bottom, that's why the arrows are big, smaller on the top, and of different widths as well. And you have to create a set of firing rates such that when convolved with those twitch responses, you get a simulated force that's as accurate as possible. And something I won't show here, but is important to these simulations, is this idea that it is good from an across-trial variability perspective when you can to use many small motor units rather than a few big ones, because if you use many small ones, the noise will cancel out. And so you can get the mean right either way, but you'll have less trial-to-trial variability if you make heavy use of the small motor units. So you, of course, have to do this for a wide variety of different force profiles, and we can pretend to be the brain. We can solve this optimization problem in this toy example. It's not easy, but it's doable. And these are, in fact, the optimal recruitments of 5 motor units with the twitch responses that you see up above in these 4 different circumstances. So this is actually, at least in the simple case, what would really be optimal. And I want to draw your attention specifically to this case here. Notice that early on, when forces are more slowly changing, motor unit 1 is the most active. Later, motor unit 4 is active. They're active with somewhat different phases, right? Being optimal requires a lot of work, right? You've got to solve a difficult problem, potentially producing a fairly complex output. And the idea historically has been, don't do that, right? It turns out that for slowly changing things, the optimal solution is very simple, which is just tend to mostly use your small motor units when you can, and then start using the big ones only when you have to, right? And so that's the idea behind the size principle, and it's thought to maybe even be sort of created via the biophysics of the motor neurons themselves, with the smaller ones being more excitable. such that all cortex or anybody else has to do is give a common input that says, give me this much force. The small motor units get activated first, then the medium ones, then the big ones in the way that most of us are familiar with. And of course, this goes back to Henneman, who was very firm in his belief that this was obeyed no matter what the source of the drive to the motor neuron pool, which supported the idea that it's an intrinsic property perhaps of the motor neurons themselves, and lots of work on this topic. So importantly, there's not just a theory about what motor neurons do. It really has implications for how we think about descending control, right? And that's been part of the reason why it's been considered so important, right? So that's put nicely here by Karp and Walpole, who say that the size principle frees higher senders from the need, right, to deal with each and every motor neuron, right? And allows them to just send down a simple command. Similar idea is expressed in principles of neuroscience. And again, the key thing that's thought to be sort of broad importance is this is, again, relieving the cortex from the burden of controlling a lot of things downstream. It allows it to just say, hey, I want this much force, and then everything else is taken care of, right? And so very specifically, it's said that the brain shouldn't, doesn't, and can't selectively activate specific motor units because that would be too much work for it, right? So subtle point is none of this really has to do with the size principle per se. The point is just that there needs to be some, I mean, maybe there's some muscle where you would want the size principle to run in reverse for some reason of optimality, and maybe evolution would make it work that way. The important thing is just that there's some rigid order, some rigid rule telling you that for a given command, how active each motor unit is going to be. And that's this notion of rigid control. And the idea of rigid control is that you've taken degrees of freedom away from cortex or other descending controlling areas, right? So you may have a lot of muscles and a lot more motor neurons, but you're going to have these relatively simple descending commands, fixed recruitment, probably according to the size principle, and then that gives you relatively few degrees of freedom that you actually have to control. But, you know, what if it doesn't work that way, right? Let's consider something kind of insane, right? Maybe you actually have very high dimensional descending commands. Maybe you actually have very flexible recruitment, and maybe you're controlling a lot of degrees of freedom. Why? Well, you know, if you want to be optimal across a broad range of situations, and presumably evolution does, you're going to want to do that. And although the benefits of taking away degrees of freedom are sort of clear if you're like a 70s or 80s era roboticist, you know, this picture makes sense. It's not clear it really makes sense from the standpoint of the brain or motor cortex. We don't know how it's solving the problem of motor control. For all we know, it's easier for it to find a solution if it has a lot of degrees of freedom. We don't really know what's easier or hard from the standpoint of cortex. And certainly the anatomy, especially in primates, can support it, right? So we have these very large corticospinal tracts, again, in a human, over 1 million axons. And because they're only controlling about 50,000 motor neurons, that's 20 to 1, right? Like it would not be a problem if you wanted it to exert relatively detailed descending control. So why isn't this a solved problem? And part of the reason is during most standard laboratory tasks, rigid and flexible control actually make very similar predictions. So take this one standard task, you have to produce an increasing ramp of force, then hold it, then slowly let go in a decreasing ramp. And what we know from the simulations we just did, size-based recruitment is optimal, right? So flexible control predicts size-based recruitment because it's optimal, so the brain should do it that way. Rigid control predicts the same thing for a completely different reason. It's because it's optimal. Evolution has made you do it that way and you have no choice, but it's still the exact same prediction. So many experiments have examined recruitment empirically during slow ramps. A few have during faster ramps or things approximating steps. Very few, however, have in this sort of interesting circumstance where you really expect to see big departures from rigid control if you're capable of doing so. So break this down a little bit. Let's compare these two situations here, and I'm going to stretch things in order to put them on the same time scale so that you can appreciate that the force on the right is much faster. The simulations we run predict that actually in both cases you should get size-based recruitment in both of these circumstances, right? There's not really, the two hypotheses don't actually make different predictions. And that's consistent with dogma, right? The idea is that the order of motor unit recruitment doesn't change with contraction speed. And a number of different groups have come to the same conclusion that although you might think that you would selectively use your fast twitch fibers for rapidly increasing things, that doesn't appear to be the case. And here's just while we're citing canon, here's yet another quote reinforcing the same point. And actually, this is exactly what we would expect, because optimality doesn't require a departure from rigid control in this case. So you wouldn't expect to see one, but it does in this case. So, what about historically, what's happened with sinusoidal forces? Have people seen things like this where one motor unit is more active first when things are slow and another is more active when it's fast? Maybe. So during slow and fast cycling, something like this seems like maybe it's happening, but it's been observed very indirectly by analyzing the frequency content of bulk EMG, not by looking at actual individual motor units. There's then this one, literally one example from a paper on lengthening contractions, where the motor unit at the bottom is quite active for the fast lengthening contraction, and the motor unit in the middle there is not very active, potentially suggesting some form of flexibility. But most reviews on this topic say, well, even if this does happen, it's sort of specific to this weird thing of a lengthening contraction, right? A lengthening contraction is the muscle's getting longer, but you're resisting the force that's making it long, but not enough, so it's continuing to lengthen. And maybe something special is thought to happen during lengthening experiments, but generally speaking, the size principle is still supposed to hold. So we're going to investigate whether that's actually true, right? We want to know, is recruitment rigid or flexible? And if it is flexible, is that cortically accessible? So we need to develop a task that allows us to record in the situations where the two hypotheses make different predictions, right? We need to make high quality intramuscular recordings, and then we need to be able to perturb activity in cortex and see whether or not we can selectively recruit different motor units. So really, Naja deserves full credit for developing this task. I thought we should do something simpler and easier. He thought we should do it the right way, and he won out. So this is our Pac-Man task, which some of you may have seen at this point. The monkey doesn't even move. He just exerts an isometric force against an immovable handle, and he only ever presses forward. So the whole task is just press forward in different amounts. And the stronger the monkey presses, the higher Pac-Man rises. And then as he releases the pressure, Pac-Man immediately falls. So Pac-Man's height is directly proportional to force. Why would the monkey make Pac-Man go up and down? Well, to eat dots, of course. And here, a really nice feature of Naja's design is he can arrange the dot pattern any which way in order to request exactly that force pattern be produced by the monkey. So we now have the ability to request the precise force patterns that we think might be revealing and might distinguish between these two hypotheses. So here you'll get to see it in action. At the top, you'll see Pac-Man. At the bottom, you'll get to hear some motor units being recorded. You'll also get to hear some clicking of the juice as he performs the task. There's a slowly increasing ramp. That's a 1 Hertz sinusoid. It's just a high static force. And as you can see, he gets very good at this. I don't know if you could actually hear, because that doesn't always work over Zoom, but it doesn't really matter whether you could or not. The key thing is to be able to see what a good job he's doing of tracking all of these different forces. These are all the different forces that Naja presented him with. And I just want to point out just how good a job he's doing of tracking these forces. So the target is the dash trace there. The black is on average, and then the gray is the individual trials. So he's really quite consistent and quite good, which is hard when you're getting up to 3 hertz, because 3 hertz is really at the limit of monkey and human ability. It's hard for us to really accurately move any faster than that. And then while he's doing this, we get these very nice motor unit recordings that we can then sort. And, these days, this is much more recent data, we would do this differently. We would, use Sam Sober's lovely arrays, which can provide you with stable recordings, even in a moving animal. But at the time, those didn't exist. So we used these modified fine wire electrodes that we modified to be more selective. We put many of them very close together within the muscle so that many of them are recording the same motor units. And the Pac-Man task was critical for achieving stability because the animal doesn't move at all. It was also critical because it allows us to measure force very, very accurately, and that force is created by a relatively small number of muscles. And I'll say even with Sam's arrays, the Pac-Man task is still really nice from a stability standpoint, even though it's not nearly as necessary as it once was. So the key, of course, is we can take these voltage traces, we can sort them into spikes. We do so based on the fact that across our 8 channels, each individual motor unit has a very unique spatio-temporal pattern of spikes. They're kind of complex because that's what motor unit spikes look like, because there's many fibers going at once. And then even in situations like this one on the right here, you can see at the right of this trace, there's a lot of motor units that are active at once. We're still able to, for example, say, this unit number 12, there it's spiking, there it's spiking, there it's spiking, and Nodge's algorithm is capable of, even when 2 units spike and superimpose on one another, because that superposition is linear, it's relatively easy to undo. So even in a circumstance like this, where there's a fair bit of overlap, we can still do a good job of spike sorting. So what do the data look like? So let's first consider this situation here where we're going to give different patterns of forces. So as predicted, and by predicted, I mean as predicted by what would be optimal if you could do it, recruitment of motor units does indeed depend upon the force profile. So here, and I'm just showing 4 channels out of 8 for simplicity, and I'm just showing a brief 100 millisecond little snippet here from the end of one trial during that little period that's in gray there. So that's when he's just hitting near maximum force on this ramp. And you can see 1, 2, 3 lovely little motor units that are nice and active with very clear, easily sortable spikes. During this period here, which is of the same duration, things are on a slightly different scale above, he's ramping up to roughly the same force that he hit on the left, but now he's doing so very quickly as he's tracking a 3 hertz sinusoid. And you can see quite a different set of motor units is active, right? Yeah, 3 and 2, and 1, they're still in there, but so's number 5, so's number 6. And that's true not just for that trial, but that's true across all the trials we looked at. So here's just a couple more examples. The things on the right are the ramps, are the sinusoids, the things on the left are the ramps. And you can see there's these additional motor units that become particularly active, often before even the motor units that were active during the ramp. So let's concentrate just on 2 motor units, this one here, number 5, and this one here, number 3. 5, we're going to make blue, 3, we're going to make red. So during the slow ramp, the red motor unit, you can see is quite active, and the blue one mostly isn't. But then during this fast sinusoid, the blue one is more active than the red one, and the red one's actually slightly less active than it was during the ramp. So clearly inconsistent with rigid control. Here's a second example. The red motor unit here just really loves this ramp. It's presumably a small motor unit because it just turns on right away and stays on. And the blue motor unit really doesn't care about the ramp at all. But once things get to high frequency during the chirp, the blue one really turns on and the red one's actually turning off. So again, this is a level of flexibility incompatible with rigid control. So to drive that home, let's think about, well, what is it really that rigid control predicts, right? It predicts that there's a single common input that's driving all of the motor neurons, but each individual motor neuron is allowed to respond to it in its own separate way, captured by these link functions here. And that it's the common input pushed through these link functions that determines the actual firing rates that we observe. And indeed, we often do observe firing rates that are quite consistent with the predictions of rigid control. And in particular, we see that especially when forces are changing slowly. I'm going to express now the same idea in a different format. So on the left, we're going to have a more complicated input. Okay, it's going to get pushed through the two link functions in the middle, and we're going to get the firing rates on the right here, right? You might initially be tempted to think this is a violation of rigid control, right? Because the red unit is more active at slow forces, and the blue one is more active for rapid forces, but that's really not true, right? They can both be accounted for simply by the same input being pushed through different link functions, right? So this would be consistent with rigid control. And an easy way to see that is actually to plot things in a state space format. So we're going to plot motor unit 1 versus motor unit 2. And now, even though we're moving quickly in time, at least in the state space, there's this very simple monotonic manifold. Sorry, somebody didn't like something and gave a big thumbs down. Oh no. What does that mean? All right, well, we won't worry about it. The idea is that even though things kind of look complex when plotted versus time, when plotted in state space, there's just this, all the data should lie on this monotonically increasing manifold. And so we can use that to quantify things. So here, imagine that we have two data points, RT and RT prime. In this two-dimensional motor unit firing rate space, we know that these 2 points are consistent with rigid control because, as in that dashed line, right, we can draw a monotonic manifold, and as long as activity lives on this line, it can pass through both of those points, right? On the right here, once we pass through this first point, RT, we're stuck in these green zones, right? There's no monotonic manifold that can then allow us to get to our RT prime down there. And so we know we have an error in this case of at least 8 spikes per second. It might be more, but we know it's got to be at least that. So we can use this fairly conservative measure to sort of run through all our different data sets on all our different days and our different conditions and ask, how bad does displacement get, right? And here there's very little displacement because the data is mostly consistent with rigid control. In this case, there's going to be a lot of displacement because the manifold occupied by the 4-second ramp is very different from the manifold occupied during the high-frequency sinusoid. So we've got a big displacement. Over here again, right, the ramp, motor unit 329 just doesn't care about the ramp, but it's actually very active during the higher frequency parts of the chirp. And so we're going to get this big displacement. A very subtle point is some of these loops are actually consistent with rigid control if they can be accounted for by a small latency shift. And we take that into account in the analysis. So it only counts as a displacement if it can't be corrected by a latency shift. So just to quickly sum up, this is just a way of summarizing across all our different sessions, with each session being a black line there. We always get a lot more displacement when we observe all the conditions versus just the ones like the slow ramps that are supposed to be, and as far as we know, are consistent with rigid control. I'm going to, in the interest of time, skip through that. There's another metric we can use. I think it's worth going through how we quantify this. We call this dispersion. We sweep a line down, a diagonal line, because a monotonically increasing manifold can only cross the diagonal line in one place. And then we ask, do the data cross it in 2 places, right? And they do here, but not by very much. So that's consistent with rigid control, more or less, right? Whereas here we get large amounts of dispersion. because we cross that diagonal line in a couple of different places. And here you'll see the same thing happens again. And this is just another way of being able to quantify across all of our different sessions on different days. And indeed, we see a lot more dispersion when we include a variety of different force profiles versus just the ramp. Okay, so what about muscle lengths? So it's potentially interesting because if the deltoid is in this position and you're pushing up, it's shorter than if you're in this position and pushing forward. So that's exactly what Naj is going to do during this task. He's going to go in and move the entire physical apparatus like that in order to shorten or lengthen the deltoid. And different fibers have different overlaps of the actin, myosin motors and might be differently useful at different muscle lengths. And that's something that maybe recruitment should leverage. And indeed, as you'll see, it really does. I'm going to show you just 2 channels out of 8 because that's all you need to see this effect. You get very different recruitment patterns at the two different muscle lengths. So, for example, motor unit 4 is very active when the muscle is long. When the deltoid becomes short, it's not nearly as active. Whereas motor unit 5 really wakes up and fires away. And just looking at a few more examples, here's a case where the red motor unit is very active when the deltoid is long, and the blue one is not, but when you shorten it, that activity pattern reverses. So again, inconsistent with rigid control. Here's just yet one example. Different motor units are differently active depending on whether the muscle is long or short. And I'll just rush through the quantification, but across multiple sessions, you always get more dispersion across muscle lengths. So finally, what about cortex? Is any of this flexibility accessible to cortex, right? Because it wouldn't necessarily have to be, and some of it likely isn't. I mean, you could use purely proprioceptive spinal feedback in order to get at least some of the effects that I just showed you could be obtained that way. So does cortex have any ability to flexibly recruit motor units? So I'll start. I'm trying to structure each of these 3 sections. We're on the last of the 3 empirical sections, such that you can see the effects at sort of 3 different levels, right? The individual spikes, because it's really important to know that your spike sorting is good and you're not getting effects just because of lousy spike sorting. And also at the level of then individual examples where you can see trial averaged rates, and then at the level of the quantification. So we'll start using that same pattern. We're going to focus just on these 2 motor units. The red one has a much smaller spike than the blue one, so I've made it much larger. So that's why that black scale bar is a lot larger. But they're very clearly distinct based both on their size and on their shape. And when we stimulate on electrode 23, we get only the red motor unit. We're stimulating during that green period there. You can even see a little bit of a stimulation artifact in the background. That's not noise, that's a stimulation artifact. And you get 2 lovely little spikes when you stimulate on that particular electrode. So this is just one contact on a V probe. When you stimulate 400 microns away on electrode 23, you don't get the red motor unit anymore. You just get 2 nice spikes from the blue ones. You go back to electrode 23, you only get the red one. You go back to electrode 27, and you only get the blue one. And if we look across a whole bunch of trials, we see that when we stimulate on 23, there's very little change in force. We're activating very few motor units, and so there's a negligible change in overall force. But you can clearly see this red motor unit becoming active, and there are its spikes. If we move to stimulate on site number 24, both the blue and the red unit are active, and there are their spikes. And now that we're a full 400 microns away, now we're recruiting almost entirely only the blue with very few spikes coming from the red. And of course, all of these trials were interleaved during the actual experiment. And it's also true, as it would almost have to be, given what I just showed you, that perturbations of cortical activity can perturb this sort of natural recruitment order. I mean, we sort of learned at this point there is no single natural recruitment order, but let's just say the recruitment order during a slow ramp. So the blue unit here is much more active during the slow ramp, with the red unit becoming active only at the higher force levels. But when we stimulate, even though we get just a small little negligible change in force, the only unit that we recruit is the red one and not the blue one. So very much they're being recruited out of order. Here's another example of the same thing happening here. The red unit, but not the blue unit, becomes active during the ramp. The blue unit just doesn't care at all, but the blue unit is very easy to drive from cortex, and the red unit is not active at all. And again, I'll just draw your attention to the force traces at the top here, right? We've got a lot of force here, and yet the blue unit is not active. Here we've created such a small change in force, you'd have to zoom in here to see anything at all, and yet the blue unit is very active. So it tells you that we can cortically activate just a very small number of motor units very selectively. And again, I'll rush through the quantification. But I will linger briefly on this form of quantification because it's the best and certainly my favorite. So we know that the thing on the left is probably consistent with rigid control. That looks like a classic figure from a size principle, book chapter. The thing on the right is definitely not consistent with rigid control. So how are we going to quantify this? Well, let's get back to where we were before, right? This is this premise of rigid control. There's one input. You don't know exactly what it is, but it's something kind of like force. And then there's these link functions that you also don't know, but at least we believe they're monotonically increasing. And then pushing the input through the link functions gives you the firing rates. And again, we see things that are consistent with that in the data. But we want to know whether that generally works, right? And this state space format is very useful for getting a sense as to whether models of that type are likely to fit, right? Oh, sorry, I'm getting slightly ahead of myself. The general idea here is that we're going to observe the thing on the right, right? We get to observe the activity of 2 or really, more like a dozen simultaneously recorded motor units. And we want to know, do there exist a set of link functions and some common input that could explain the things that we observe, right? And if there are, then it's definitely going to have to be the case that everything's going to have to live on one of these monotonically increasing manifolds. Like that we know for sure, with the exception of some little loops that can happen just for latency differences, right? So for example, you kind of know this is going to be consistent with rigid control, and this definitely is not going to be consistent with rigid control. But again, we want to quantify that. So we observe these rates. Depending on the experiment, there might be 6 motor units. There might be, I don't know what the max we ever got was, low 20s. And we want to learn as best we can link functions and latent inputs that could potentially explain those rates, right? And we want to find the best latent inputs and the best link functions that we possibly can, right? And if there don't exist any that can observe the rates, then and only then can we really reject this model, right? So we've got to learn these link functions, and we want to be as open-minded as possible. We want to be overly generous, right? So let's not assume that link functions look like this. Let's learn them in some way that allows them to be any monotonically increasing function with like minimal smoothness requirements, even something that we wouldn't really think was that realistic, because we are trying to be as generous as possible to the rigid control hypothesis. And again, we're going to let the latent input be anything it wants to be, right? So this is a very difficult optimization problem, but fortunately, if you're friends with the right people, it is actually solvable. You can take something like that, and you can get a model fit. And you might ask, well, how is it getting those little wiggles? Well, it's some combination of the fact that it's allowed to have some latency differences among the motor units and the fact that it's allowed to have anything it wants as the latent input and the linked function. So it can actually fit that pretty well. However, for data like this, that's real data, right? It just can't do it, right? There exists no latent input and no linked function that could possibly ever do that. And so the model just fails. And we can quantify that in the following way. We can fit only the slowly increasing ramps and compute the fit error. And keep in mind, that allows us to optimize our link functions only for the slowly increasing ramps. Now we're going to say, no, we have to fit everything. Now everybody can have their own independent latent drive, but they've got to all share the same link functions, right? So now all of a sudden the link functions that we can use during the slowly increasing ramp are constrained by the fact that the link functions have to work across all of the other conditions. And now we go back only to the slowly increasing ramp and we ask whether our error increased, right? So it's always the fit to the slowly increasing ramp where we're computing error. And we're going to compute it in a cross-validated way such that it'll be 0 on average if indeed the only error is due to measurement noise. And you can see it is basically 0 when you're fitting only the slowly increasing ramp. But once those same link functions have to work other places, now all of a sudden you're having to make compromises in those link functions, and they no longer work well for the slowly increasing ramp. And we can use the same method to show that across muscle lengths and as a function of stimulation, this model does not fit. But you'll see it actually always does fit well for the slowly increasing ramps. And I'm not showing this, it actually also fits well for a combination of slowly increasing ramps and rapidly increasing ramps. It's really once you get things like the fast sinusoids or the muscle lengths or the stimulation involved, that's when the model of rigid control really falls apart. So how many degrees of freedom do we think there are? I mean, you need more than one latent drive. In these very flexible models, we can estimate that we think there's probably at least 4. And of course, there could be many more once you include an even broader range of behaviors, right? We only explored a very small subset of all possible behaviors. So I'll finish pretty quickly, but I think it's important for me to say that cortical activity is rich enough to support this kind of detailed control if it needed to. And I want to say that because there's this idea often that cortical activity in motor cortex is low dimensional. And dimensionality reduction is a very useful tool, but that's not the same thing as saying activity is low D. And in fact, the very first application of PCA and dimensionality reduction to motor cortex data, the whole point was that the data is actually quite high dimensional. In those old data sets, we were estimating, at least 30 dimensions. And actually, I'm going to sort of skip through that. And what I want to say, and this is leaning on some of Abby Russo's data, and if you haven't seen this, I'm going to rush through it a little fast. But the point here is that as cortical activity evolves, there is a direction in state space where if you project activity onto that readout direction, you get the activity of individual muscles, right? So it's absolutely plausible that cortex controls individual muscles, but those readout directions are small. In fact, in this particular example, we've actually expanded, Abby's expanded that vertical axis so that you can see it. And that's a common property of neural networks. That's not surprising. And if output signals are small, it means you could potentially have many of them, right? Even if most of your variance is in the top 2 dimensions, you can still have a lot of small dimensions that are contain meaningful signals that could control a lot of degrees of freedom. And we can get at this by recording neural data and asking how much diversity in the Pac-Man task is there across neurons. And to make a long story short, if you use a measure of dimensionality reduction that doesn't say how big is a signal, but it says how reliable is it across trials, across partitions of the data, and assesses whether it's real that way, you find that there's, not just 50, but potentially hundreds of reliable small signals in motor cortex, right? So in a model that only has 50, this metric plummets faster. You're going to need at least on the order of 100, 200 or more underlying degrees of freedom to account for the complexity and the high dimensionality that we see in motor cortex. So even though it is true that a lot of the variance is often taken up by 10 or 20 dimensions, there are a lot of extra dimensions that, even though they're small, are real and could potentially support this kind of detailed control. And I think I'm going to, even though I really like this last slide, in the interest of leaving enough time for questions, I'm going to skip skip that and ask you guys your thoughts, opinions, questions, and so forth.
Great. Thanks, Mark. Are we all, yeah, we're all on the screen. Any questions for Mark?
First of all, who's Hector Cho? Do we know that?
I think Hector is Mark's student, right?
Okay.
That's your student, Mark? Can you hear us?
Yes. Yes, he's not a random interpolator or spy.
He
Okay.
must have been the thumbs down person then.
Oh my God. Yeah, who was the thumbs down person?
You might have just interpreted that we sometimes it happens that, Zoom, it happens to me constantly that Zoom thinks I'm doing something and.
Oh, like it looks at your gestures.
Yeah.
Gotcha.
Oh,
All right.
I didn't know it could even do that, but I'm actually feeling good there was only one thumbs down in the middle. I thought I'd get a couple of slides, so I'm all right with that.
Recognize my eyebrows when you said manifold instead of function.
Okay, sorry, wait, break that down for me there.
I said when you said manifold instead of function. I mean, it's what you mean is a monotonic function between the two.
No, I mean a manifold.
Thumbs down.
I don't think that's a wrong term.
Oh, probably.
I mean, it's keep in mind it's not in 2D, right? It's really in 200D, right? So it's not, we're not saying motor unit 2 is a function of motor unit 1, right? Right, we're saying all the motor units are a function of an underlying drive that you don't get to see. So that, if you want to call it a function, call it a function in that domain. But in the state space, right, if you don't know anything about any underlying latent variable, in the state space, the object I'm describing is a one-dimensional monotonically increasing manifold.
Okay, that makes sense.
Yeah.
I have a question, Mark, about the possibilities of controlling the descending drive versus the cortical outputs, changing the link functions.
Oh, that's a great question. Yes. Yes, you're right. We fundamentally can't distinguish between those two things. And at some point, maybe it almost becomes philosophical whether there is a distinction. Because that's a very good point, right? There's two ways you could get flexibility. One is you could have multiple descending drives, each with their own link functions. If they all share the same link functions, it doesn't work, right? So they've each got to have their own link functions. Or you could have a single descending drive, but then control over the link functions. I think at some point, once the model starts to fail, it's questionable whether or not you want your new hypotheses to use the same framework as the old hypotheses.
Yeah.
I'm not sure you don't, but I'm not sure you do either. So, but yeah, it's a very good point, right? You could easily imagine that it would be more natural to explain things in terms of changes in the link functions.
Or maybe, I was thinking more even whether one could go after some kind of, cell type specific fantasy where you have, the more direct cortical motor neuronal projections doing obviously the input, but the kind of, cortical spinal in general, making changes in the link function simultaneously.
Yeah, I mean, I think I don't know that I could defend this, but at least in my head, changing link functions to me sort of seems like something that slow neuromodulation, changing link functions that's a very natural way for me to think of things. But so much of this is happening on timescales that are way too fast for that. So I tend to think of it in terms of multiple, descending drives as the easier way. But either way, you're looking at more degrees of freedom than you had before. Yeah.
Questions for Mark? Yeah, Murda?
Yeah, I have a question about the interpretation of the stimulation experiments in general. So even assuming that we have like many dimensions available in the cortex, we have like a specific mapping between cortex and a spinal cord, I assume. And when we stimulate, it can go in any direction, presumably out of that like very high dimension manifold. So isn't that guaranteed that if you stimulate enough, you would eventually see something that the spinal cord or the motor neurons do not do or show even like in the normal conditions, meaning non-stimulation cases?
No, it's definitely not inevitable. I mean, put it this way, suppose that suppose that that rigid control, that the size principle were really enforced by the biophysical properties of the motor neurons themselves, right? Then it's not at all clear that you could ever violate that, no matter what descendant command you sent, right? You might get very unnatural things that the system would never do, but they would all still obey the size principle, right? Because it is enforced below the level at which you are stimulating. So, and in fact, the claim of Henneman was absolutely that if you stimulate cortex, if you stimulate brainstem, if you stimulate by getting a reflex, that you always get the same the same recruitment order. So I think both logically and historically, no, it would not be inevitable. At the same point, I would completely, endorse your point that it's not like we think that the patterns of activity we're sending down are natural patterns. In fact, we very much don't. Our hope really is actually to generate a lot of very unnatural patterns in order to see what the system is capable of doing. Of all the forms of flexibility it has, I assume it uses a relatively small number of them during most of daily life, because it wouldn't be optimal to be you got a lot of degrees of freedom. It doesn't mean you want to use them all, right? But you might still need that many degrees of freedom such that you can be optimal in any situation.
Yeah. Hi. So one quick question that I have is, do these pattern of flexible recruitment has anything to do with the history of the force activation? For example, like in the centrifugal case, for example, there might be a decrease and then a rapid increase. I wonder if it's controlled, that, for example, you compare the slowly increasing force with the case that they both come after a rapid decrease, for example.
Yeah, it's a good question. I'm not completely sure of the answer. We didn't see any obvious history effects, with one exception, which is sometimes you get these, especially following stimulation, you'll get things that are pretty clearly the result of a persistent inward current. And I actually had an example in there and pulled it out in the interest of time. So we're careful to be sure that none of the effects that we're showing you are things that could be explained by persistent inward currents, although we do occasionally see that happen. And occasionally you see that happen too, even during normal behavior, right? In the usual way, there'll be some small motor unit that turns off late for the ramp down in sort of the classic PIC way, but that's not where most of the violations come from. We did also create a version of Josh Glazer's model. It was his model that did the fancy fitting at the end. We created a version that is allowed to have some internal dynamics and therefore some history, in order to basically say, does one latent plus a bunch of history effects, can that rescue the model? And it couldn't. You could always claim that model wasn't expressive enough, because once you're in that domain, it's sort of impossible to be expressive enough. But we were never able to sort of explain things via history effects. But that said, I wouldn't rule them out. And depending on their nature, they could be either evidence for or against flexibility, right? There might be some boring history effects like PICs, but then also some history effects that actually are like things flexibility would want to do. So yeah.
Paul. Hi, Mark. Paul Gribble.
Hey, Paul.
Hi. So I have kind of a two-pronged question. You kind of cast the main thing that distinguishes different motor units is their size. We also know that motor units in different parts of a muscle can be recruited differently depending on the task and the configuration of the limb, for example. So I kind of wanted to ask how that fits in. But
yeah. And so,
in the context of the idea that is there any organization in cortex, anatomical organization, or as Andrew said, cell type organization that could provide an anatomical substrate for controlling different flavors of motor units, big, small, and different parts of muscle?
Yeah, it's a great question. I think there's sort of two prongs to the first prong of your question, right? And the second prong is sort of a forward-looking prong, like, given these multiple forms of diversity, including probably a bunch that we don't know or can only guess at, like, for example, overlap in the actin myosin motors. There's, generally speaking, muscle fibers are diverse little things in terms of which genes are expressed inside of them. So there probably is a lot of diversity there. And it may even be that the sort of speed, fast twitch versus slow twitch, may not even be the most important kind. I mean, we sort of got moderate violations of rigid control with different speeds. We got huge violations of rigid control with different postures and with stimulation. So I think you're right that there's a lot of forms of diversity, and it may even be that the one that most people think is the most important may not be. There's another prong there, which I'm not sure if you meant or not, but it's an important one, so I'm glad you brought it up, which is there's a known violation of the size principle that most people insist is not actually, it's sort of technically a violation of the size principle, but it's not considered one, which is that for, and I'm pretty sure this is what you were getting at, like, for example, in the case of the bicep, it has 3 different mechanical actions, right? I mean, it obviously it flexes the elbow, but it also is involved in pronation, supination, and actually also to some degree with motion across the motion of the shoulder. And those 3 different actions involve different parts of the bicep to different degrees, and they each have their own size principle. So it's as if the size principle gets sort of, the historical idea is that the size principle is just repeated 3 times in a slightly different way for each of those actions. And yes, the size principle still holds, but like Bawa, for example, is very explicit about this, but it should be thought of as holding within the group of motor units that are most responsible for a given action. So historically, that's been thought to be part of rigid control, but I think, maybe in the future, some things that were thought to be, maybe we'll realize that you actually do have a lot more flexible control over different parts of the muscle than we realized you did. I don't know. It's a reasonable question. Yeah. Oh, but actually, sorry. The most important thing I should say is that is not where any of our effects come from, right? We made really sure that the monkey is always using the same mechanical action, right, across all of these different situations. And we record not only for muscles that are what are called, I think they call them multifunctional muscles, but also muscles that classically are not. So we know that is not the source of the violations that we're observing. They're not like, they can't be explained away in that way. Yeah.
Thanks, Mark.
John?
I have an existential question.
Yeah.
Traditionally, a lot of the time we know our models are good fits to neural data because of, explaining a lot of variance. That's a lot of way that's been characterized. But based on what you just said about motor cortex, there might be a lot of very small dimensions that are really the most important control dimensions, where you could have potentially tons of models that seem to fit the data, but actually miss these very important dimensions. So going forward, how do we know when our models are right and when they're not, if we're relying largely on variance?
Yeah, it's a really good question, right? And I think this is not the only example that I have. There's other examples where and it sounds like you might have a few examples of your own, where small signals are the important signals. And, I think tangling was one of the first things that really drove that home for us. I mean, we knew that in a lot of these recurrent networks, the output dimensions tended to be small, but, we imagined, oh, well, if you regularize them, they'll at least be sizable. They'll at least be, 10 or 20%. And then we realized that actually it can be very optimal to keep them small, right? The little bit of noise that you get in your output doesn't really hurt you, the noise you really want to avoid is noise that perturbs your dynamics and makes you unstable or causes you to make internal errors. And that trade-off means that you make the dimensions that reduce tangling very large, such that even if your muscle trajectory is not a big circle, you really want your neural trajectory to be a big circle. And you can look back at the rotational trajectories during reaching. It's just a specific instance of you want a big looping trajectory in order to keep tangling low, and then your readout is something that's close to orthogonal. And so yeah, you're absolutely right that it's going to be tricky, and it's probably a reason why neuropixels are going to be very important, is because we cannot assume that the top 8 or 10 dimensions contains all the interesting stuff. In fact, pretty soon I think we'll have good explanations for what happens in the top 8 or 10 dimensions, and they'll become boring. And it'll be what happens in all of those other smaller dimensions. It's like that's where the real, that's where the real meat will be. You'll be like, oh, of course the top 2 dimensions are a big circle. That has to be for tangling. That's boring, right? Like what are my other dimensions doing? Yeah.
It's 2:00. Any more questions for Tony?
So there are known descending pathway, including the cortical spinal, including pathway and retrospinal pathway, and for monkeys, spinal pathway and afferent input to the common motor neurons. So do you think really kind of every kind of the control dimension is quite distributed in kind of every motor neuron is controlled by every kind of common the different input, or not common input, but it's kind of somehow between the intermediate of the two kind of extreme of spectrum? What do you think about it? So, because there are still already kind of the different kind of source of input to the motor neurons.
Yeah, it's a really good question. I think I don't have an intelligent opinion. I mean, I guess I would guess that most motor neurons will be drivable from all sources, but maybe not to the same degree. But it is a good question. I mean, the thing I always find striking is just how few motor neurons you've got, right? Yeah. And a medium-sized muscle only has a couple hundred. And for a lot of the movements you're making, you might only be using a third of those. So you're like using 60 neurons to like, so my guess would be that most of those neurons can be activatable by all of the descending pathways that you mentioned, including the reflexes, but maybe not in the same way. Yeah, it's a really good question.
Thank you.
Time for one more if there is one. No. All right. Awesome. You want to?
Just quickly, Mark. From the model fitting, you get these. If you fit the model with multiple descending commands and multiple link functions, you get like 3 or 4.
Yep.
Are those? Can you get your head around those? Can you look at those and say like?
That's a great question. Josh Glazer is working on that as we speak. I'm not sure. I should check in with him and see what his recent progress has been. The short answer is so far no. And part of the problem is that, to really have a decisive test, we had to be very generous to rigid control, right? We had to say, look, you can pretty much have any link function you want as long as it's monotonically increasing. It can be something pretty monstrous that nobody would think was realistic, that, looks like a staircase or something. But as long as it's monotonically increasing, you're allowed to have it. So my suspicion is that makes it very hard to then get interpretable latents, right? Because some of the work that would be done by the latents can now be done by these unrealistic link functions. And so what we needed in order to reject rigid control was to be very generous with what the link functions could be. In order to aid interpretability, we need to now say, no, those link functions, they need to be more realistic, right? Like, they need to look more like a sigmoid or something that we would all agree is realistic. And Josh is trying to do that. And the hope is, as you said, that we will get latents that are then more interpretable. Maybe one latent that's controlling the slow part and some other latent that then kicks in and does, the sorts of things that are in your head. I would hope we would see, but we haven't had success yet. But Josh is smart. Maybe he will.
All right,
Thanks.
let's thank Mark again. Awesome.
Thank you, guys.
We appreciate it. See you soon. Awesome.
Yeah.
Bye.
It happens to me in Zoom like constantly, that suddenly
Yeah.
something will like, confetti will fall down, or there'll be a thumbs up, or.
Oh, with the
It must
recognized
have been
gesture.
gesture.
No, but it